In this session, I will be discussing about enzyme inhibition and competitive inhibition. Objectives of this session are: at the end of the session, the learner shall be able to define what is inhibition, then categories of enzyme inhibition and competitive inhibition and its applications. Let us first take up definition of enzyme inhibition. Enzyme inhibition is the substance which binds enzyme. Here note, it is not substrate. It is what I am telling is substance. Substance which binds enzyme and brings about a decrease in catalytic activity. A decrease in catalytic activity. If substrate binds it, increase in catalytic activity. Substance binds it, decrease in catalytic activity. Substance is nothing but the inhibitor. Okay, I hope it is clear. Then, what are the categories of enzyme inhibition? The next objective is categories. Next objective is categories. Okay, what are the different categories? Okay, let us see. Okay, this is the one. What are the different categories? Okay, what are the different categories? Here we say enzyme inhibition. Enzyme inhibition. Okay, the first one is reversible. The first one in these categories, the first one is reversible. Reversible. The next one is irreversible. Irreversible. The third one is allosteric. Allosteric. This is the first one, second one, and third one. Okay, what is reversible inhibition? Let us know what is reversible, okay, inhibition. Let us first discuss about the reversible inhibition and its various categories. Here, inhibitor binds non-covalently. Inhibitor binds non-covalently with enzyme. Inhibitor binds non-covalently with the enzyme, and this enzyme inhibition can be reversed. Enzyme inhibition can be reversed. Okay, reversed if the inhibitor is removed. If the inhibitor is removed. Inhibitor is removed. So here we have studied about the reversible inhibition. And reversible inhibition, inhibitor binds non-covalently with enzyme, and enzyme inhibition can be reversed if inhibitor is removed. Let us talk about the various categories of reversible inhibition. Various categories of reversible inhibition. Okay, the various categories of reversible inhibition are, let us see here again. Okay, this is the one. Then again, three categories are there. Three categories are there. Okay, the first one is competitive. The first one we say competitive. The next one is non-competitive. The third one is uncompetitive. The third one is uncompetitive. Competitive, okay, non-competitive, and third one is uncompetitive. Okay, these are the categories of reversible inhibition. Let us take up the competitive inhibition. Okay, competitive inhibition. In the next session, we'll take up the non-competitive and non-competitive. Okay, what is competitive inhibition? Okay, inhibitor, that is I, it has structural similarities with substrate. It is not structurally same. Please remember, it has got the structural similarities. In another way, we can say inhibitor is substrate analog. Substrate analog. Okay. So, because of the structural similarities, inhibitor competes with substrate for active site on the enzyme. Okay. So, in the case, what happens? There is a formation of enzyme inhibitor complex. Enzyme inhibitor complex. So, it cannot undergo catalysis. And then, so no product. So, that is what happens. So, what happens? The enzyme is occupied with inhibitor. Enzyme is occupied with the inhibitor. Let me give the diagrammatic representation. I'll write the enzyme molecule. This is enzyme. How it will form the complex with the substrate and how it will form with the complex with the inhibitor, I will explain. So, let me first take up the substrate. Okay, take up the substrate. This is the substrate. Okay, and this one is the inhibitor. This particular one is the inhibitor. This is inhibitor. Okay, now let us see how the complex formation is taking place in both the cases. Okay, in both the cases, how the complex formation is taking place in both the cases. Here there is a perfect complex. So here we can say enzyme substrate complex. Here you can see here. Then again I am writing. Okay, here it is. Okay, then what I do here, it is with the inhibitor. The complex formation has taken place. Here it is, the complex is enzyme and so inhibitor complex. Here enzyme with the substrate complex, here enzyme with the inhibitor complex. Let us see here what happens in these circumstances. In case of enzyme substrate complex, enzyme comes out. This is enzyme, and so there is a formation of a product. The formation of product formation has taken place. Here, what happens in this case? Enzyme inhibitor. Here, there is no further reaction. Enzyme inhibition is taking place. Inhibitor can occupy the catalytic site, but there is no catalysis. There is no product formation. Let me represent in another way. This is enzyme. Okay, here one side with the substrate, another side with the inhibitor. Okay, then what will happen? Enzyme substrate, enzyme inhibitor. Then finally what will happen? Enzyme plus product. Here it is, no catalysis. There is no catalysis which is taking place. So, what is the possibility of these complexes? The relative concentration the relative concentration of substrate or inhibitor determines whether it is enzyme substrate or enzyme inhibitor complex. Enzyme substrate or enzyme inhibitor complex. Okay, if more substrate concentration is present, then there is increased enzyme substrate complex. If more substrate is present, so enzyme substrate complex formation will take place. Enzyme substrate complex formation will take place. If more inhibitor concentration is there, then more enzyme inhibitor complex formation will take place. Let us see the substrate concentration plot. Substrate concentration plot. Let us draw the substrate concentration plot, how the substrate and inhibitor show. Okay, then let us see here. This is reaction velocity. Here it is substrate concentration. Let us draw a curve here. Here it is. This is without inhibitor. This is without inhibitor, and this one is with inhibitor. This is without inhibitor, and this one is with inhibitor. This one we can say without inhibitor. This is with inhibitor. This is with inhibitor. There is no inhibitor here. There is a presence of inhibitor. So here we can say V maximum. Here we can say of V maximum. Here it is Km. Okay. See this graphical representation. Okay. I will explain to you what happens when enzyme substrate complex formation taking place in the absence of inhibitor. So it achieves the maximum, V maximum. In the presence of inhibitor, in the competitive inhibition, V maximum can be achieved, but more substrate is required. The concentration of substrate shall be more. That is the reason when you see the enzyme velocity, reaction velocity, in the absence of inhibitor, the Km is low, the affinity is more. With less substrate concentration, V maximum can be achieved. Whereas in the presence of inhibitor, Km is high. The reason being is, if it has to achieve the V maximum, more substrate concentration is required. That is as simple as that. Then we can also represent in the form of the Lineweaver-Burke plot. Let us see here. So this is how we draw the Lineweaver-Burke plot. Let us see here in the presence of inhibitor, in the absence of inhibitor. This is in the absence of inhibitor. Here it is in the presence of inhibitor. Then, so what I can simply draw this one is, the first one is here no inhibitor. Here, so there is a competitive inhibitor. So here it is 1 by V0. It is 1 by S. So this is intercept of the y-axis is 1 by V maximum. They are reciprocal. Okay, reciprocal plot. If we extend this one, if we extend this curve, okay, to intersecting the x-axis, here it is, here it is. Let us see, this is 1 by km minus 1 by km. This is also minus 1 by km. Here, in the presence of competitive inhibitor, okay, km is increased. Here km is increased. Okay. Then, whereas in the absence of inhibitor, so km is less. That means affinity is more. This is the how we can show the effect of inhibitor in the substrate concentration plot and also in Lineweaver-Burke plot. Let us discuss about the application, okay, application of competitive inhibition with examples. Okay, applications of competitive inhibition with examples. Okay, so let us take up the examples one by one. Examples one by one. The first one is methotrexate. The first one is methotrexate. Okay, so it is a structural analog. Structural analog of dihydrofolate. Analog of dihydrofolate. Okay, so methotrexate is the structural analog of dihydrofolate. It is an anti-cancer drug. Anti-cancer drug. Okay, so which enzyme is inhibited here? Enzyme inhibited is, okay, enzyme inhibited is dihydrofolate reductase. It makes the active form of folate, dihydrofolate reductase. Okay, so by inhibiting this enzyme, what are the consequences? Okay, synthesis of, okay, purine, the nitrogenous base, and pyrimidine decreased. Purine and pyrimidine decreased. That leads to decreased synthesis of DNA. Okay, when there is a decreased synthesis of DNA, then what happens? Cell division is hampered. Cell division is stopped, hampered, slowed down. Okay, that is the how it is an anti-cancer drug. The second one is sulfonamides, antibacterial drugs. Sulfonamides. Okay, so it is, sulfonamide is an analog of, structural analog of para-amino benzoic acid. Para-amino benzoic acid. It is required in the synthesis of folic acid in microorganisms. Okay, enzyme inhibited is enzyme inhibited is enzyme inhibited is thyroid synthetase. Okay, so what happens? Once this enzyme is inhibited, what is the consequence? Synthesis of folic acid in microorganisms. In microorganisms is decreased. What is the consequence of this one? So it inhibits growth of microorganism. It doesn't have effect on human cells because human cells, they take the ready-made folic acid which is supplied in the diet, whereas bacteria, they synthesize the folic acid in their cells. For that, para-amino benzoic acid is required. But when you give the sulfonamide antibiotic, so it is a structural analog, so it inhibits the thyroid synthetase, so the bacteria cannot synthesize folic acid. Okay, that is how the infection is controlled. Then third one is isoniazid. Isoniazid. So it is analog of, structural analog of nicotinamide. Nicotinamide, structural analog of nicotinamide. Okay, it is anti-tubercular drug. Anti-tubercular drug. Okay, anti-tubercular drug. Okay, so it inhibits synthesis of nicotinamide adenine dinucleotide. So ultimate effect is decrease growth, inhibition of the growth of microorganism, tubercular bacilli, microorganism. Okay. Then the next one is dicumarol. It is analog of vitamin K, structural analog of vitamin K. Okay. Which enzyme is inhibited? The enzyme inhibited is epoxide reductase. Epoxide reductase. What is the role of dicumarol? It affects the vitamin, decreases the vitamin K cycle or stops the vitamin K cycle. So it affects coagulation. Affecting the coagulation process. Okay, so because of the application of dicumarol, what happens? So prothrombin time is delayed. So prolonged clotting time. Okay. Next is the statin group of drugs. They are anti-hyperlipidemic drugs. So structural analog of HMG coenzyme A. The enzyme inhibited is HMG coenzyme A reductase. Hydroxymethyl glutaraldehyde, okay, coenzyme A reductase. By giving the statin drugs, okay, cholesterol synthesis is decreased. Once the cholesterol synthesis is decreased, that leads to the decreased serum cholesterol. There are many other drugs are there. Others are, okay, ethanol in methanol poisoning. Okay, then ibuprofen, anti-inflammatory drug. Okay, ibuprofen, okay, it is anti-inflammatory drug. So the use of competitive inhibitors in medical practice is immense. There are many drugs. The mechanism of action is through the competitive inhibition. In this session, we have discussed about the enzyme inhibition and competitive inhibition. Let us summarize. Enzyme inhibition is substance by binding enzyme decreases catalytic activity. Categories: reversible inhibition, irreversible inhibition, allosteric inhibition. What is reversible inhibition? The binding between enzyme and inhibitor is non-covalent. If inhibitor is removed, enzyme activity is regained. What are the categories of reversible inhibition? Competitive, non-competitive, uncompetitive. So let me summarize competitive inhibition. Inhibitor has structural similarities with the substrate. Substrate analog. Inhibitor is substrate analog. Okay. Then, if enzyme inhibitor complex formation takes place, no catalysis is taking place. No catalysis is taking place. So, diagrammatic representation, enzyme and substrate. Enzyme substrate complex, then enzyme and product. Okay, here if inhibitor binds, which is a substrate analog, enzyme inhibitor complex, no product. Let us see substrate concentration plot. V maximum can be achieved in the presence of inhibitor if excess substrate is supplied, whereas affinity is decreased, that is Km is increased. The Km, when competitive inhibitor is present, it is more. If you compare with there is no inhibitor. If you take the Lineweaver-Burke plot, here also you can see here, the Km is increased in the presence of inhibitor. Let me summarize applications with the examples. Methotrexate, it is anti-cancer drug. It is an analog of substrate dihydrofolate. Okay, enzyme inhibited dihydrofolate reductase. Okay, so because of this one, purine pyrimidine synthesis is decreased, DNA synthesis is decreased, leading to the stoppage of cell division or decrease in the cell division. Sulfonamides, antibacterial agents. It is an analog of the substrate para-amino benzoic acid. Enzyme inhibited steroid synthetase, and due to this one, folic acid synthesis is decreased in microorganisms and growth is decreased. Okay, then isoniazid, anti-tubercular drug. Okay, analog of nicotinamide, enzyme inhibited PLP dependent enzymes. Okay, NAD synthesis is decreased in microorganisms, so growth is affected. Okay, dacumerol, anticoagulant, it is an analog of substrate vitamin K. Okay, enzyme inhibited epoxy reductase, and due to this one, vitamin K cycle is hampered and leading to the prolonged clotting time. Okay, statin drugs, anti-hyperlipidemic drugs, it is an analog of the substrate HMG coenzyme A. Enzyme inhibited hydroxymethyl glutaryl coenzyme A reductase. Cholesterol synthesis is decreased, serum cholesterol levels are decreased. Other inhibitors are ethanol, it is a structural analog of the methanol. It is used in methanol poisoning cases. Ibuprofen, it inhibits the cyclooxygenase. Ibuprofen, it inhibits the cyclooxygenase. It is an anti-inflammatory agent. These are the various aspects of enzyme inhibition, including the competitive inhibition. Okay, thank you very much.